Hello, my name is Buning and I'm from Fluidem. Welcome to my poster on expanding lifestyle barcoding applications with mass cytometry using cadmium and platinum labeled antibodies. Allow me to do a quick introduction on our technology. Our technology is mass cytometry, or also known as CyTOF, which is based on cytometry by time of flight, which utilizes metal tech antibodies for single-cell profiling to look at different markers that is expressed on the cells. This technique enables comprehensive analysis of your samples in a highly multiplex manner where you can stain your samples with a large panel of antibodies consisting of more than 50 plus markers easily, as this method is not constrained by the compensation limitation of fluorescence-based cytometry. We have recently introduced new metal isotopes to be tagged with antibodies, and with this new introduction, it has enabled mass cytometry to expand the multiparametric characterization of single cells to 57 different parameters. These new metal isotopes are 7 cadmium isotopes and 4 platinum isotopes. Essentially, with these new isotopes, this means that you can do more comprehensive analysis in a more easy and flexible manner. Previously, we have demonstrated that cadmium labeled antibodies can be efficiently used for live cell barcoding of PBMC, your peripheral blood mononuclear cells, with antibodies targeting the pan-leukocyte marker CD45. As live cell barcoding do not require fixation, a major advantage of this approach is that it could preserve the ability to stain fixed sensitive epitopes. Cell barcoding maximizes the performance of mass cytometry by enabling sample multiplexing for high parameter single cell analysis. If you look at figure 1 here, barcoded samples can be stained together in a single tube, eliminating staining variability between samples and reducing antibody and reagent use. Acquisition is also faster and requiring only a single run of the multiplex sample. With cell barcoding, you can achieve higher sample throughput for scaled-up experiments and improve data consistency for large studies. In these studies, we have expanded the live cell barcoding approach by staining PBMCs with a combination of mixed metal isotopes that includes cadmium, platinum, and yttrium in a 7-3 format to allow for multiple samples analysis of up to 35 samples in a single go. We use the live cell barcoding method to barcode the samples and then followed by antibody stainings using our MaxPar direct immune profiling assays, or in short, MDIPA. The MDIPA is a ready-to-use drag-down 30 antibodies panel optimized for deep immune profiling of either human whole blood or human PBMC samples. The 30 marker panel is shown here in figure 2 together with the workflow of the panel. Paired with the MaxPar PathSetter software, the software enables a quick within 5 minutes automated analysis workflow that allows for automated identification of 37 different immune population in a highly reproducible manner. Jumping to the method section, this plot shows the method for the mixed metal 35 live cell barcoding. The representative of the cadmium and platinum and also the 89 yttrium isotopes used in this study here is described and shown in figure 3a here. Also, the unique combination of the barcode scheme via the 7-3 format for the metal mix that allows for 35 samples barcoding is shown here in figure 3b here. The staining procedure is shown in figure 3C here, where we first take PBMCs and then perform live date staining with our cell ID intercalator rhodium. And then after live date staining, half of these PBMCs was stained with 209 bismuth CD47, which is a marker found on all leukocytes. This step was carried out as a control to assess for any intersample combination that may occur due to sample pooling and subsequent multiplexing. So after this, the cells were allocated into a 35 individual tubes containing the method labeled anti-CD45 antibody barcode mixture. And then after that, once the samples is being barcoded, each tube may contain approximately 2.5 million cells each, will then combine into a single tube and then stain with antibodies CD66B and also CD19, both with individual different metals. The CD19 is consisting of 142 ND, and while the CD66B contains 162 dy metal. All antibody staining were stained according to our Fluidan MaxPa cell surface staining protocol with fresh fix. The multiplex samples in a single tube was then acquired on the Helios instrument running on the Cytometry software and debarcoded using the Zunder single-cell debarcoder. On the results section here, you could see the debarcoding results. Here we use a setting of 0.03 for minimum barcode separation based on the generated curve, while for the maximum Mahalanobis distance, the MD, were used as the default value as there were no major outliers was observed. So looking deeper into the debarcoded data here, it reviewed consistent data quality. Here, in A, the histogram revealed clear signal for barcoded versus non-barcoded channels, while in B, it shows similar results using heat map format with clear, strong expected positive signals, which is aligned with the barcoded scheme used. In the C section here, the iridium DNA2 versus the 142 ND-CD19 biaxial plot revealed minimum differences in the population frequencies across the barcoded samples. And lastly, over here in D, the Disney plot of all the 35 barcoded samples here shows that all clear. There's a clear separation of all the 35 different barcoded samples, where you can see all the individual population of all the barcoded samples here. Here's the method for the live cell barcoding followed by MD-IPA panel staining. Whole blood samples from 2 healthy donors were first stimulated with PMA ionomycin for 2 hours prior to antibody staining, as shown here in figure 4b. Antibodies were stained according to the MD-IPA user guide together with the unique combination of 3 platinum CD45 antibodies barcode that were added to the samples during sample staining. After sample staining, samples were then combined into a single tube after red blood cell lysis step. A non-barcoded control from the unstimulated donor 1 was also included and stained with only the MD-IPA panel for comparison. All the samples consisting of the multiplex samples together with the non-barcode control were all, as mentioned earlier on, combined into a single tube and proceed with the remaining staining protocol followed by acquisition on the Helios mass cytometer. The results section for this setup is shown here. we use a minimum barcode separation of 0.3 based on the generated curve. While for the MD value, we use 20 to remove any outliers. Overall, you could see clear separation between the positive, shown in yellow histogram, and also the negative, shown in black histogram signal. Also, you can see here the expected positive channels align with the barcoding scheme used. Diving deeper to the results and looking at population frequencies between barcoded versus non-barcoded samples, the results show here that the live cells barcoding does not affect cell frequency, shown here in figure 8a, as both show comparable frequencies here with minimal differences observed. Over here, you can see the population frequency change in the CD8 T cells stimulated with PMA ionomycin, indicating that the panel and the software could detect these changes in samples. So as conclusion, using live cell barcoding labels leukocyte with anti-CD45 antibodies could help you to increase multiplexing power to 35 samples or more. In this study, we did a 7-3 format. If you use the total of 11 metal isotopes from our newly introduced metals isotopes consisting of the 7 cadmium metals and also the 4 platinum metal isotope and use a 11-3 format, you can essentially multiplex up to 165 different samples in a single go. The added flexibility in the tag selection for a combination of cadmium, platinum, and yttrium isotopes empowers few users to pursue live cell barcoding studies with extensive customization of antibody panel. The live cell barcoding can also be expanded to other applications, including whole blood from healthy donor stained with our high-dimensional antibody panel, such as the MD-IPA panel here. So with this, I thank you for your attention and happy to take any questions. Thank you.